So we're going to talk now about active transport. So again, just putting you back in the big picture. Oops, sorry, let me get the pen going here. So you have transport, you have passive transport, and the other type of transport is active transport. There's two types, primary active transport and secondary active transport. So what makes active transport active is that it's going to require an input of energy. This is because it's moving the opposite direction. Instead of going from high to low, it's when things are moving from low concentration to high concentration. This is called going against the gradient. And as I mentioned in a previous video, this is analogous to going uphill. So to get a ball to go uphill, you have to push on it. You have to put in energy to get it to go uphill. So again, sort of the same thing, we're going to have to put in energy. There's two types of active transport. Primary active transport is going to directly use energy. So this is going to require a specific direct energy source, ATP. Secondary active transport is going to be an indirect use of energy, and it's going to have to use a different molecule and a whole extra step, and we'll see the example when we get there. So we'll start with primary active transport. This is carried out by molecules referred to as pumps. So pump references the use of energy. It's going to burn fuel in order to move solutes against their gradient. That fuel is ATP. So breaking down ATP. ATP is a molecule that can get broken down into two pieces. We'll get into the names of this molecule and these different parts in Chapter 6. For now, we'll just leave it as we're breaking down ATP, and coming out of that is a bunch of energy that can be used by the cell to do work. So energy to do work. And in this case, that work is going to be the movement of solutes. So on this side of the membrane, I have low calcium. On this side, I have high calcium. But the cell wants to move calcium from here to here. And so that's moving against the gradient, that's pushing it uphill. So it will take the calcium, load it into the carrier protein, and then move it to the other side. But in order to make that movement, it's going to have to burn fuel. And so it uses ATP. So it's pumping calcium against the gradient from low to high concentration. The sodium-potassium ATPase, so that just means it moves sodium and potassium and it burns ATP. So big fancy name that means it moves sodium and potassium and burns ATP. This one is a classic pump that's classic to learning about biology, partially because it helps to maintain the electrochemical gradient we were talking about in the previous slide. So remember I said that overall this inside of the cell is negative, the outside of the cell is positive. This is accomplished by having differences in ions. So the sodium-potassium pump is going to export, so it's going to send out positive charges. So it's going to send out 3 positive charges, and it's going to bring back in 2 positive charges. But notice that if I do that, it's actually 1 less positive charge on the inside. So if I send out 3, I remove 3, bring in 2, I've subtracted 3 and I've only added 2. That's leaving me with -1 on the inside. Okay, so that's helping maintain the cell as negative. So the way that it works is that it loads in 3 sodiums from this side. So let me sketch in the concentrations here to start. So on this side I have low sodium. On this side I have high sodium. On this side I have low potassium. On this side I have high potassium. So sodium is going to get moved from here to this side, and potassium is going to get moved from this side. So for both sodium and potassium, this is active transport. They're getting moved against their gradient. So that's going to require energy. So we can see that I'm going to have to use ATP here to accomplish that. So 3 sodiums load into the enzyme, then the enzyme flips. So again, sort of using my hand, oops, there's 3 sodiums. The 3 sodiums come in, and then it flips, and the sodiums leave. So that was the pen. Now the potassiums come in, and the protein flips back, and the potassium comes out. And then it's ready for the whole thing to happen again. So let me show you that one more time. So it starts out like this. The sodiums load in, and 3 of them load in. Once 3 of them are loaded in, it's going to burn energy. So there's me burning off my lunch, flipping my hand. If only it was that easy. And now the sodiums can come out, and that makes space for potassiums to come in. So here comes the potassiums, and then that causes the protein to flip back to its original shape, and then the potassiums come out. And then the whole thing can repeat again. And so again, what happened there was 3 sodiums left and 2 potassiums came in. All through this protein flipping up and down, changing shape. So facing down, then facing up, bringing in the potassiums and facing back down. So we'll watch an, whoops, sorry about that. We'll watch an animation here, so I have to get into slideshow mode for you to see it.
That is driven by the
Oops, and let me start that over for you.
The sodium-potassium pump is an active transport mechanism that is driven by the breakdown of ATP and works through a series of conformational changes in a transmembrane protein. 3 sodium ions bind to the cytoplasmic side of the protein, causing the protein to change its conformation. In its new conformation, the molecule becomes phosphorylated at the expense of a molecule of ATP. The phosphorylation induces a second conformational change that translocates the 3 sodium ions across the membrane. In this new conformation, the protein has a low affinity for sodium ions, and the 3 bound sodium ions dissociate from the protein and diffuse into the extracellular fluid. The new conformation has a high affinity for potassium ions, 2 of which bind to the extracellular side of the protein. The bound phosphate now dissociates, and the protein reverts to its original conformation, exposing the 2 potassium ions to the cytoplasm on the inside of the cell. This conformation has a low affinity for potassium ions, so the 2 bound potassium ions dissociate from the protein and diffuse into the interior of the cell.
Now that had a little more detail than you need to worry about, but I think the key is that you really saw well how the protein moved. It was open on one side, then it was open on the other side, and that facilitates the movement of sodiums from one side and potassium from the other side. All right, so we'll talk now about secondary active transport. So secondary active transport is still movement. Oops, sorry about that. Is still movement from low to, or sorry, rather high to low. No, I was right the first time. Low to high concentration. It's still going to require an input of energy, but it's going to be an indirect use of ATP. So let's focus on this part of the figure first. What I have is on this side of the membrane, I have high H plus, low sucrose. So 2 different solutes at different levels. On this side, I have low H plus, high sucrose. And what the cell wants to do is move sucrose from low to high. So this is a food source, so it's trying to get the food into the inside of the cell. Whoops, I apologize here. Oh no, never mind. So it's moving low sucrose to high sucrose. Okay, so again, that's active transport. The cell wants to do this because this is a food source. And in order to accomplish this, it's going to catch a ride with hydrogen. So on this side of the membrane, I have high hydrogen. On this side, I have low hydrogen. So if I move this way for hydrogen, it's passive transport. It's diffusion. So let me just over here draw this a little more simplistically. So for H plus to move from top to bottom is going from high to low for H plus. To go top to bottom for sucrose is to go from low to high. They're both going to get moved together by the same protein, so this is a symporter. The same protein will do this. If I do this at the same time, I can use the hydrogen ion to move the sucrose. So remember I said imagine going down the gradient is going downhill, right? This will happen on its own. The sucrose is just going to catch a ride with it. Because this wants to happen, if you will, the sucrose can just catch a ride with it and move this direction, even though it would normally be uphill for it. So it's having to use energy, but the energy is coming from the hydrogen ion going downhill. So that's an indirect use of energy, but still having to use it. The reason why it's indirectly a use of ATP is because as the hydrogens flow this way, I'm eventually, in order to keep the gradient, going to have to send those hydrogens back out. So the hydrogens have to basically make a circle. So this is for the hydrogens. As they come in, they're moving sucrose, right? And that's why the cell wants to do this, is it's moving sucrose. It doesn't really care about hydrogen ions. It just wants to bring in sucrose, and it's using H+ to do that. Well, in order to keep using H+, I have to send H+ back out. If I send H+ back out, that's moving from low to high. Guess what? That's also active transport. So now it's going to have to use ATP. So a second protein actively pumps hydrogen against the gradient using ATP. So let me summarize this one more time. Over here, the same protein is going to move hydrogen down its gradient, and that provides the energy for the sucrose to move against its gradient. So it's a symporter. It's moving both of them at the same time, but for one, it's down the gradient, and for the other, it's against the gradient. This is going to have to use ATP because as those hydrogens flow in, they're eventually going to have to be pumped back out to keep this process going. So over here next to it, in a different protein, hydrogen ions have to be pumped from here back out, and this is going to use ATP. So it indirectly provides energy by burning that ATP. So that is secondary active transport.